Good morning, everyone. I am Professor Guihua Hou from Shandong University Basic Science of Medical School. Today, we will start a new course called Advancing Molecular Image. This course mainly faces some students from abroad or graduate. This course contains 8 lectures, well teaching by several teachers or doctors coming from Shandong University Basic Science of Medical School and affiliated Qilu Hospital. Today, I will give you the first lecture, General Introduction. In my course, I will talk with you about 5 different parts. Part 1, Concepts and Features of Molecular Image. Part 2, Molecular Recognition Theory. Part 3, the Probe of Molecular Image. Part 4, Classification of Molecular Image. Part 5, Application and Development of Molecular Image. Now, let me introduce the first part, Concept and Features of Molecular Image. Actually, image could be divided into 2 classifications. First is anatomic imaging. It relies on tumor size, shape, density, example, X-ray diagram, computed tomography. You can be used to do this anatomic imaging. It measures response by changes in size or density. It's anatomic imaging. Actually, it's a structure change. Secondary is functional imaging. Functional imaging relies on in vivo disease-related biology, perfusion, metabolism, molecular features. Actually, it measures response by changes in functional or molecular processes. So you know the imaging divided into 2 types, anatomic imaging and functional imaging. Molecular imaging belongs to function imaging. So you should know molecular imaging not show the anatomic change. It will show the function or metabolism change. So the first important thing you should know about molecular imaging definition. What is molecular imaging? It uses imaging technique to display specific molecules at tissues, cells, or subcellular levels to reflect changes at molecular level in vivo, and to conduct a qualitative and quantitative studies of the biological behavior in imaging. About this definition, you should know 2 important things. First, you use the imaging technique outside the body, outside the living body. And the second thing you know is measure the change on the cellular or subcellular level, or we can say it's on molecular level. Molecular imaging techniques directly or indirectly monitor or record the spatial temporal distribution of a molecular or cellular process for biochemical, biological, diagnostic, or therapeutic application. These 4 molecular imaging definition. What is molecular imaging? Now we want to know about the principles of molecular imaging. Actually, molecular imaging is to combine molecular biology and other basic theories such as microbiology with radiological imaging science. for example, CT, for example, MR, for example, nuclear medicine. This radiological imaging science to form a new branch of medical science. So you know the principle of molecular imaging is combined two things. One is basic science, molecular biology, immunology, microbiology, and this basic series with clinical used radiological imaging science to form a new branch, like these figures showed. For radiology, nuclear medicine, for ultrasound detection, for MR, for CT. For basic science, you know the immunology, you know the microbiology, and microbiology, and this is very important. So we can see from this molecular imaging, you can see the figure, or we call pictures. Molecular imaging technique has three key factors. First, you should prepare a high specific molecular probe. Second, you should have an appropriate signal amplification technique to apply the signal derived from the probe. The third, you should have a detection system that can obtain a high resolution image sensitively. This image showed where the probe is and how the quality and quantity of unknown probe binding target molecules. Molecular imaging integrated genetic information, biochemistry, and new imaging probes into a human body or some living organisms. They use them to label a target. To enlarge the target by the molecular imaging techniques, detect it by precise imaging technology, and display the living body through a series of imaging post-processing technology. The purpose of molecular imaging is shown the biological process at a molecular and cellular level, transformed to subclinical diagnosis and treatment of disease. For this picture, you may see the first part, basic research. You found disease-related protein, cell receptor, DNA and RNA change, or some special protein change. And then you can find them in vivo image. So you can use the imaging machine to show the DNA, RNA sequence, to see the special protein, to see the receptors or other disease-related protein. Molecular imaging combined microbiology, biochemistry, data processing, nanotechnology with imaging processing and other technology. Because of its high specificity, high sensitivity, and high resolution of image, it can really provide a qualitative, positioning, and quantitative data for clinical diagnosis. Thus, molecular imaging is no longer a single technological change, but an integration of various technology. It's combined several advancing technology to form a new branch. So about the process of molecular imaging. What kind? How it's working? First, for molecular imaging, we should approve a specific molecule expressed on the tissue. Example, a kind of receptor. And then we need to prepare a probe targeting this molecule and verify this probe in vitro, usually with a cell line cell culture in vitro. Then you should verify this probe useful in animal model, usually for some disease model. And finally, you should transform this technology, this probe, to clinic to do the disease diagnosis or to evaluate the therapy. Now I will talk the main features of the molecular imaging. First, molecular imaging can provide in vivo molecular gene information and even multi-molecular interaction information, non-invasively, minimally, and repeatedly. This unique and authentic individual information is premise of the individualized medical treatment. Second, molecular imaging is an important technology tool for basic research and an important bridge for the transformation of basic research result to the clinical reaction, such as usually we call translation medicine. The third, molecular imaging support targeting therapeutics and can be used to evaluate therapeutic effect in real time at a molecular level. If the patient receives a chemical therapy and how this drug is useful for this patient, you can use this technique, molecular imaging, to show whether the patient received this therapy is effect or no effect. Then you can adjust the treatment method. The fourth, molecular imaging showed obvious and huge advantage in the whole process of the drug development, include new drug discovery, research drug metabolism distribution, secretion, absorption, or for the target organ or tissue. And these are four main features of molecular imaging. We can conclude these important features in four. The first one, very important, is non-invasive. It's minimally at the lowest level for patient is invasive. So it's non-invasive. You don't need to cut. You don't need to hurt. You don't need to let patient to receive any harmful. So you just give an injection, and then you can get the result. And the second important feature is in vivo. Remember, molecular imaging is used in living body, patient, alive organism, not dead. It's alive. And the third is real time. That means the patient lie on their checking bed. You can see the image to know whether the patient somewhere has the receptor changes. It's real time. And you can see the qualitative or quantitative change. They have this, yes or no? How much they change this disease and where the disease. So real time. The last is visualization. You can see it through the image. You can see the patient receptor change or some special protein change and some other DNA, RNA change in a living body, not for outside the body. So molecular imaging is very interesting and very important. Okay, that's all for this part. Thank you.